All right, now to part two of this paper, Scaffolding and Natural Selection. We'll look at section 3 of Rainey's solution, which he called ecological scaffolding. Rainey has introduced his notion of ecological scaffolding in 3 recent publications, though precursors of the idea can be found in his earlier work. Above, I briefly sketched the idea as the exogenous imposition of Darwinian properties by the environment. And this is largely how Rainey et al. 2017 defined the concept, seeing the environment as a crucial and neglected factor in the major transitions by exogenously imposing, quote, Darwinian properties on otherwise unwitting particles, page 104. Rainey himself sees his contribution as involving both a succinct statement of the essence of the circularity dilemma, which he thinks has been ignored by many in the field, and a straightforward solution, that is ecological scaffolding, which simply follows from the former clarification of the problem. A more precise definition of the solution has been given in Black et al., where they maintain that the, quote, Darwinian properties can be exogenously imposed in such a way as to cause lower-level entities, for example cells, to become passive participants in a selective process that occurs over a longer time scale than the time scale over which cell-level selection occurs and is part of a larger collective-level entity, quote end. As I see it, Rainey's notion of ecological scaffolding is essentially a combination of these 3 ideas. One, an older suggestion by Wilson and Sober in their reviewing the superorganism, that selection on groups could take place in virtue of the environment rather than properties internal to the group. 2, a general growing awareness of the importance of ecology beyond population structure within the study of major evolutionary transitions. And 3, the exploding usage of the term scaffolding across the sciences to explain complex phenomena. But the idea of ecological scaffolding is not merely intended as a theoretical contribution to debates in the philosophy of biology. While his career began in experimental biology, Rainey developed an early interest in the more theoretical questions in biology. His research has long been concerned with the question of how multicellular organisms evolved from free-living, single-celled organisms, serving as a paradigm example for an experimental biologist becoming engaged in a debate that was once the almost exclusive domain of evolutionary theoreticians and philosophers of biology. Rainey's introduction of the term ecological scaffolding builds on decades of ongoing experimental and theoretical work on the evolution of multicellularity, was explicitly intended to be operationalizable and guide further empirical work. The primary example used for ecological scaffolding is an experiment by Hammerschmidt et al. 2014. In this experiment, they attempted to test Rainey's hypothesis of Jesus propagules in terms of the new terminology, a scaffold for group-level reproduction, variation in heredity through a single-cell bottleneck. The bacterium Pseudomonas florensis is propagated in test tubes with a nutritious proof. Mutations quickly arise that lead to adhesive cell glue production that bind cells together. The cost of glue production is twofold. Firstly, the cost of producing the glue itself, and secondly, the costs of life in close proximity, as daughter cells cannot detach themselves from their parent. However, some such mutant assemblies are able to survive by taking over the interface between air and breath, attaching themselves to the walls of the microcosm, without which these non-buoyant mats would sink. They occupy a new niche, rich in oxygen, a benefit that outweighs the costs. And here, a quote, a long one from the paper, describing a figure that's omitted here for copyright reasons. Please look at the actual paper. Quote, a putative life cycle for mat-forming bacteria. We start with a single bacterium given in blue, capable of producing an extracellular adhesive. 1. It reproduces at the interface between liquid and the air, in the case shown, starting at the inner surface of a glass tube. Daughter cells stick together because of the adhesive they produce. 2 and 3. The resulting mat spreads over the liquid surface as a single layer. Due to prime access to oxygen, a robust mat forms. Mutations generate sheets. Green cells that do not produce any adhesive polymer and grow faster as a consequence. These sheets spread like a cancer in the mat and contribute to the collapse of the mat. Because the sheets do not produce the adhesive, they are liberated from the mat upon collapse. Back mutation from one of these sheets to a mat-producing cell completes the life cycle. Of course, we do not imagine such a life cycle playing out in an environment where only a single mat can form, like a singular tube. Rather, the back mutants from the liberated sheets could establish mats in different locations from the parent mat. Here, the cell type leading to the death of the group also leads to its rebirth. The sheets amount to propagules, germline, arising de novo from the mat-forming soma of an incipient multicellular individual. And reigning care, 2010. In virtue of the access to oxygen, these cooperative groups achieve access to a limiting resource, taking over the entire surface. At some point, however, mutations lead to cheating cells living within the mat without producing glue themselves. These cheats prosper as they have access to oxygen without the costs of glue production. The mat becomes heavier and loses structural integrity, until the lack of cell-cell glue leads to the doom of the mat, that is, the extinction of the protogroup organism. Of course, it is not meaningful to speak here of a Darwinian individual in its own right, unless it has some means of reproduction. Godfrey Smith. But Rainey and Kerr suggest that the very cheats that spell doom for the mat could also be its savior, by functioning as a sort of propagule, forming a new mat and thereby the beginning of a new life cycle. Hammerschmidt facilitated a grand-scale experiment showing that a cheat-embracing regime in which a new life cycle begins with a cheating cell is able to decouple the fitness of groups from the fitness of individual cells. The lower level units become de-Darwinized, as Godfrey Smith might put it, by becoming subservient to the life cycle of the collective unit. The first steps towards multicellular organisms have been taken in the lab, successfully testing hitherto philosophical ideas. As I previously argued, some may legitimately criticize the artificial nature of this experimental setup. The point here is not to claim that there is some deep metaphysical divide between artificially induced selection and real ecological scaffolding in nature, but rather that whereas it is comparatively easy to exogenously induce Darwinian properties in the lab, aggregates of lower level units in the world outside the lab rarely meet the conditions of evolution by natural selection because they lack heritable fitness differences. One might thus criticize the Hammerschmidt et al. Work for lack of a plausible story of how their model could map onto the real world, but this would be to misunderstand the paper. Similarly to mathematical modelers, experimental biologists such as Rainey are often asked to provide us with a detailed story that maps the artificially induced selection onto natural selection operating in the real world. Such a requirement forces the abstract and generalized explanations presented in papers reporting the data of an experimental model to narrow to particular target systems. This then misinterprets the very point of the original paper, which was to provide a very generalized and idealized framework that would have to be filled out for the particular contexts we're interested in. When examples and narratives for such general explanations are provided, they're mostly suggestive, providing one possible way in which the explanation could be realized, not how they must be or how it could actually happen. Or how it actually happened. The lack of specific cases is thus not a flaw, but a feature quite similar from modeling in biology. The economist Robert Sugden has called such models fittingly, quote, explanations in search of observations, quote end. I suggest the same applies to laboratory experiments exploring general mechanisms operating in living systems. Rainey et al. Offer a supporting analysis, quote, the overarching goal of much experimental evolution is to simplify in order to understand processes too complex to fathom in real-world situations. Quote end. Indeed, in Black et al., they further idealize from their already fairly abstract example to provide a general and simple model that highlights the important role of ecological factors as possibility proof for the imposition of Darwinian properties through properties of the environment, which has been corroborated in the experiments. Nevertheless, even if it is possible to scaffold Darwinian properties, we will still require plausible scenarios of how this could have happened outside the laboratory. In order to illustrate the importance of ecological scaffolding, it is necessary to provide examples of how something analogous to the artificial scaffold, such as Beakers in the Hammerschmidt et al. Experiment, could have been hijacked by natural selection. In Rainey et al., They provide one plausible story of how multicellularity could have evolved in the evolutionary past, or future for that matter. It is here that I introduce the idea of ecological scaffolding, using the case of reeds as an example for the imposition of Darwinian properties. Block quote here. Consider a pond with randomly placed reeds, each of which constitutes a scaffold around which a mat can form. Mats eventually collapse and go extinct due to their increasing mass, but provided a mat can re-establish at the original reed or around one or more reeds, then a process akin to collective level reproduction occurs. With this comes the possibility of a Darwinian process at the level of mat collectives. Quote end Paul Rainey et al. Instead of the artificial setup, this scenario depicts a plausible scenario for how all three Lewonian conditions can be scaffolded at a collective level through patchily distributed resources at reeds and means of dispersal that allow for competition between mats. This provides an interesting analogy to natural selection. Darwin's theory of natural selection has provided an explanation that drew on artificial selection to explain the appearance of purpose in the natural world, the diversity of living beings, and the fit to the environment. Much the same applies for ecological scaffolding, by externally inducing Darwinian properties to unwitting particles. Instead of intentionally placed scaffolds for a specific purpose, ecological scaffolds are blind and random occurrences in the natural world that can nevertheless serve a purpose in the evolution of Darwinian properties. Providing the following illustration, Rainey et al. Argue that ponds could constitute a plausible scenario for ecological scaffolding outside the lab. As Figure 2 illustrates, reeds could possibly enable the creation of a sort of proto-life cycle by exogenously imposing the necessary and sufficient conditions for evolution by natural selection. Given the right ecological conditions, mats then become subject to selection themselves. Take these reeds, figure omitted for copyright reasons again, but look at the actual paper. A long block quote here. Pond scum acquires Darwinian properties via ecological scaffolding. The illustration shows 6 reeds in a pond. Surrounding each reed is a set of different microbial mat-forming types. Reeds are sufficiently widely spaced as to prevent confluent growth of mats, thus ensuring variation at the level of mats. Consider that a yellow mat occupying the reed marked with the solid arrow collapses. Death of this mat provides opportunity for the birth of a new mat, provided there exists a means of dispersal by biotic or abiotic means between reeds. In this example, cells from the red mat recolonize the vacant red reed. The dispersal and recolonization event is akin to a mat level reproduction, and because the cells founding the new mat came from the old mat, the offspring mat resembles the parental mat. There's heredity. Mats begin to take part in the process of evolution by natural selection by virtue of Darwinian properties that are exogenously determined. Additionally, selection sees 2 timescales, the doubling time of individual cells and the doubling time of mats. Continued selection under such ecological conditions allows the possibility that Darwinian properties could become endogenized. That is, they come to be determined by the activity of the collectives themselves with no need for scaffolding. An early stage might be the evolution of a developmentally determined life cycle. Quote end Rainey et al. 2017. So take these reeds away and the mats no longer have the relevant Darwinian properties for natural selection to occur. Now it should be clear why they don't just use the widely used and imprecise metaphor of environmental scaffolding to refer to any sort of environmental supporting relationship, such as caporal et al. Instead, opting for the previously vacant term ecological scaffolding, which they intend to have a more precise meaning. Reproduction can take place via biotic and abiotic means. While reeds are biotic compounds of the environment, nothing of course prohibits the occurrence of something roughly analogous to reeds as an abiotic component of the environment playing the same role. In the experiment, this was the beaker, or rather the surface of the beaker, at the interface between the broth and air. Consider, for instance, the case of stones in a pond peeking out of the water surface as an abiotic scaffold. Though perhaps a less realistic scenario, we keep in mind that evolution of multicellularity has independently taken place at least 25 times. See Smith and Sasmari, 1995. Hence, We should not be surprised that the actual evolution of multicellularity in eukaryotes may have been a fortunate interplay between extremely unlikely environmental conditions and the right mutations occurring in the required order. Proto-life cycles acting as marginal Darwinian individuals, far from being stable, are constantly threatened by extinction. And this is a fate we should expect almost all proto-life cycles in evolutionary history to have suffered. The multicellular lineages we observe today does represent only those stable enough to persist. Another figure omitted for copyright reasons. Again, look at the actual paper. Long quote here from Rainey et al. Ecological scaffolding in a droplet-based evolution machine ensures droplets are units of selection. Parallel horizontal lines are walls of Teflon tubes containing an emulsion of oil and regularly spaced droplets harboring bacterial cells. A. At the start of operation, all droplets are founded by identical types. B. A period of cellular growth occurs within droplets. During this stage, mutations within individual cells arise that affect the color of each droplet. Droplets whose color is not sufficiently bright are marked for extinction, allowing the possibility that when the contents of the droplets are diluted in order to establish a new round of selection, the brightest droplets are split into two offspring droplets. C. Selection thus works over two time scales, the doubling time of cells and the doubling time of droplets. As in figure 2, Darwinian properties are imposed scaffolded on droplets, causing droplets to function as units of selection in their own right. Rainey et al. 2017. The creation of such proto-life cycles in the lab, able to increase the cell stability before they eventually go extinct, is already quite an achievement. These experiments suggest ways forward, illuminating the ecological scaffolds that may have played important roles during the evolution of multicellular organisms. As Rainey et al. 2017 note, quote, ecology is everything. The structure of the environment permits realization of Darwinian properties at a collective level, even in the absence of these properties being endogenously determined. Quote end. Use of the term ecological rather than environmental scaffolding also highlights the importance of eco-evolutionary feedback loops in ecological scaffolding that can lead to the evolution of genuine life cycles. Only in virtue of a cheating bracing regime which scaffolds met reproduction do we get a germ-soma distinction, with cheating cells playing the role of proto-germ cells, a proto-life cycle that can plausibly become a unit of selection. And with selection coming to act at the level of the mat, these exogenously imposed Darwinian properties can plausibly come under developmental control, that is, become endogenized. The second example Rainey et al. 2017 offer is perhaps less obvious. Unlike the previous thought experiment, this one does not have a straightforward real-world analogy to the role of reeds in the development of mats and the construction of a building via scaffolds. The focus here is the general phenomenon of the exogenous imposition of Darwinian properties. Nevertheless, it could be made into a model for the evolution of multicellularity if each droplet was founded by a single cell, which could be instantiated through pores and alkaline vents. Dolcier et al. Have developed a mechanistic model with this motivation in mind and show that egalitarian transitions can occur through ecological scaffolding. Consider the standard conditions for natural selection listed in section 2. Phenotypic variation, differential fitness, and heritability. Upon closer inspection, it turns out that all three conditions for natural selection are here themselves being exogenously imposed by the experimenter. And without the experimental scaffolds, the Darwinian properties at the level of drops would cease to exist. In order to get distinct groups of individual cells, the setting above depends on, quote, enclosure within a droplet surrounded by oil, Rainey et al. 2017, enabling the evolution of group-level variation. The oil here can be seen as a natural border for multicellular organisms, one that is abiotically scaffolded. If individual cells can freely move from one group to the other, group level properties become diluted. While not strictly necessary for selection to occur at the level of groups, some form of boundedness is required to have a natural selection conquer, or as Godfrey Smith might put it, de-Darwinize the lower level units in favor of the group. Boundedness strengthens the role of higher level selection, since phenotypes have a higher correlation between generations. thus opening the possibility for group-level beneficial adaptations at the level of individual cells. The scenario explored in Figure 2 similarly requires that the reads are placed sufficiently far from each other for meds not to overlap, but close enough to enable differential reproduction. In addition, differential fitness of groups is here introduced by the introduction of an extinction process of darker groups, while the brightest groups are allowed to take over vacant niches of now extinct groups. Both differential fitness and heredity at the group level are here imposed via the experimenter. But as Black et al. 2020 demonstrate in a follow-up study, these properties could nevertheless become endogenized. I hope that this detailed analysis of Rainey's proposal and the examples of its application have given a good indication of the great potential it has for the interdisciplinary research among philosophers, experimentalists, and modelers. It's also an elegant example for how cooperation among these groups can lead to progress on a problem we have grappled with for decades. Philosophers of biology would be well advised to help practicing biologists to develop this research program further, and I hope that the next part of this paper will set the first stones for this exciting new avenue of research by attempting to make the notion of ecological scaffolding more precise, and once again extend the scope of the Darwinian model of evolution by natural selection. Now, section 4. Scaffolding and natural selection. Black et al. 2020 exemplify the idea that, quote, evolution is the control of development by ecology, and an idea famously endorsed by the American evolutionary biologist Leigh van Baalen. Ecological scaffolds are exogenously imposed and can subsequently become endogenized, thus eliminating the circularity problem for the application of natural selection to explain the emergence of Darwinian individuals at a new level. This is an exciting proposal with compelling empirical evidence for the early stages of the endogenization of Darwinian properties, that is, development which deserves further philosophical analysis. In this section, I will draw on recent work in the philosophy of biology to do just that. Offer a general, yet nevertheless simple definition of scaffolding as a distinctive phenomena in nature, and clarify its importance with evolutionary biology. This will help us to conceive of natural selection itself as a scaffolded process. A simple definition of scaffolding. To begin with, we should get a firm understanding of what scaffolding is. After all, the title of this article is Scaffolding Natural Selection. Yet this question is far from easy to answer, precisely because the term is used in a deliberately vague and metaphorical way, oftenly enough at least. I've noted that it has had an explosive growth in recent years, but its usage has varied widely due to the different areas in which it is employed. Nevertheless, substantial collaborative work across fields has already culminated in some progress, such as the volume Developing Scaffolds in Evolution, Culture and Cognition, Cabral et al., which arose from the 23rd Altenberg workshop called Scaffolding in Evolution, Culture and Cognition at the Konrad Lorenz Institute for Evolution and Cognition Research, the KLI. The prefix developing was deliberately added to emphasize the tentativeness of the project in attempting to make more sense of scaffolds across field. The volume explores conceptual and empirical questions in scaffolding across different disciplines and can here only be recommended for anyone seeking a broader overview across of this topic in fields beyond evolutionary biology. Cabral et al. Argued that, block quote, the word scaffolding is both a noun and a verb. It names a structure and a process. The common meaning of scaffolding refers to a temporary structure of platforms and poles erected so that workers can build, repair, clean, or decorate a building. Scaffolding is widespread, so widespread that its attachment to discourses in biology, culture, evolution, and human development indicates its centrality to processes of support and change of many kinds. Cabral et al. 2014. Indeed, scaffolding processes understood in this sense are ubiquitous in nature. We can distinguish scaffolds as structures from scaffolding as a process, although the distinction can admittedly get blurred. But the way they are loosely described here in terms of an environmental supporting relationship provides us with hardly any purchase on how to make them useful scientific concepts. It would turn a coffee cup into a scaffold in the same way as a beaver logs as a scaffold to build a dam. That environment always plays a role in evolution would hardly constitute an interesting or novel thesis. It is almost universally accepted. Nevertheless, the ubiquity of scaffolds will remain an important for feature of several reasons. Once we turn to the relationship between scaffolding and natural selection. The most interesting property of scaffolding in relation to natural selection is undoubtedly, as the example of Rainey's work illustrates, its alleged usefulness for explaining black boxes in traditional research paradigm. Carperell et al. Likewise point out, block quote, scaffolding is a missing concept, perhaps because its primary virtue is that it is commonly temporary. It is often external and either falls away or becomes assimilated to a part of the scaffolded relation, quote end, Carperell et al. Now, this is precisely the sort of relationship we discuss at Rainey's thought experiment concerning reeds, at which Metz could form, and his experimental work on the origins of endogenization. Here, Darwinian properties are in an important sense induced from the outside. While one might expect scaffolding to differ widely from one context or target system to another, there's a straightforward analogy here to familiar cases in engineering where the term originated. At construction sites, scaffolds serve as temporary structures to enable or at least aid the creation or maintenance of a physical structure, such as cranes and metal frames, used to construct buildings. The common language use allows for a straightforward definition once any intentional notion, such as the purpose of the scaffold, is eliminated. I propose the following working definition for scaffolding. X is a scaffold if and only if 1. X exogenously induces or supports the realization of property Y in process system Z. 2. X vanishes from or becomes part of the system, while property Y in process system Z becomes endogenized. The important notions in my definitions of scaffolds are 1. The exogenous realization of properties relevant to a process of system, such as Rainey's external imposition of Darwinian properties, and 2. The endogenization of these properties, that is, when they become part of the system itself or fall away without important properties or effects, thereby being lost. It is the second part of the definition that is unfortunately sometimes neglected, with the mere modal possibility of endogenization seen as sufficient for treating something as a scaffold. But as I shall demonstrate below, this would make the notion too permissive to be used as an explanatory concept in its own right, making it an almost trivial consequence of the fact that few, if any, processes occur independently from their environment. Given the excessive usage of the terms scaffold and scaffolding, being perhaps not always justifiable applied in multiple diverse domains, one may worry that my attempt to provide a general definition is doomed to failure, and that perhaps the only unifying usage would be a vague metaphorical usage. But if this were true, the vigor with which philosophers have joined in numerous debates, embracing the position would appear quite strange. Doubly so, because unlike traditional philosophers of science, many of these philosophers have actually used it to probe new research programs, an undertaking that can only serve as exemplary. But there is a tension in recent work that tries to 1. Highlight the ubiquity of scaffolds in nature, and 2. Use it as an explanatory concept. Scaffolding can either be understood broadly as any kind of entity standing in a supporting relationship, or as a more precise concept that relies on the eventual endogenization or elimination of the scaffold. My definition rests on the motivation that is only the latter understanding that can do genuine explanatory work. The former has far less explanatory value, as supporting relationships can be found almost anywhere, without thereby providing any further explanatory understanding of a target system. Since I am here primarily interested in naturalist conceptual engineering, see Fight and Browning 2020, rather than a mere conceptual analysis of the term, my focus is on endogenization. Unfortunately, many famous proponents of scaffolding seem to defend a much broader former notion, including Rainey. Here, one may object that my reservations against a broad usage of the term scaffolding are biased by my focus on major transitions. But as I shall argue, such an unconstrained usage of scaffolding fails to distinguish it from other processes and obscures the unique role that scaffolds play in nature. Kim Sterelny, for instance, has been one of the most fervent users of the term scaffolding, but that very feature may also explain his apparently very broad definition of the term. Consider his example of Canberra's road signs. Quote, block quote, these cue names directions to important locations, speed limits, and rights of way. They are not deceptive, they are regularly present, their content is highly reliable. This set of resources scaffolds navigation around Canberra's confusing street network, Kim Sterelny. Road signs, of course, are here to stay. When Sterelny speaks of environmental scaffolding, he uses the term interchangeably with environmental supports. Perhaps scaffolding is the more attractive name for seemingly providing a novel position, but it can also serve to create an illusion, applying a new term for a point that would otherwise appear somewhat trivial. My point here is not that the term scaffolding cannot be used in those circumstances, but rather that it would make the notion explanatorily hollow and mix it up with a scientifically useful but more restrictive application of the term. After all, the claim that many processes in the natural world, such as navigation, perception, cognition, and the development of organisms, are supported by the environment, is far from novel, nor particularly interesting. In fact, it is doubtful that anyone would object to such a vague version of scaffolding. More so, it raises the question of why the term was used to begin with if all it states is the existence of supporting relationships. Scaffolding, rather than a scientific concept in its own right, seems to be used as a mere analogy to strengthen the idea of niche construction. That is a process in which the organism alters its own selective environment to its own benefit, in both the biological and cultural realm. While there's metaphorical use, let me just drink something. Is not problematic in its own right. Indeed, can play an important role in science. My definition above is intended to provide a simple account that has the following triad of useful features. 1. It is generalizable across different contexts and domains. 2. It can be used in actual scientific work. And 3. It closely tracks common language use of the term. Caporal et al.'s volume did not attempt to provide us with a general definition, with its contributors using the term in widely different ways. Sometimes solely metaphorically, sometimes with a precise and narrow meaning, fitted to the particular target of the investigation. And this was certainly admissible in the first collected volume on the topic. The goal was to let 100 different flowers bloom. And a deliberately vague use of the term was the ideal scaffold to accomplish just that. But in order to make the notion a precise scientific concept, there's an eventual need to provide a more precise and general definition that is applicable to the myriad scaffolding phenomena in nature, with their different timescales and levels of organization. Even if such an attempt at policing the usage of the term will strike some as unwelcome. Admittedly, my definition may very well fail in due course and be replaced with a better one. Perhaps it will be demonstrated that the various phenomena conceived as scaffolding processes are too disunified as to allow for useful scientific concept. This is not too much of a problem, as it is beyond the scope of any single standalone paper to survey an entire literature and prove that the definition stretches across all important instances of scaffolding processes. Nevertheless, if such an objection can be brought forth against my constrained definition of scaffolding as a process of homogenization, it is even less likely that a much broader definition can play a useful role in science. For the present purposes of turning scaffolding from a metaphor into an explanatory concept, my general definition will enable us to think about natural selection itself as a scaffolded process, enabling us to explain the major transitions in a non-circular, yet nevertheless Darwinian manner. Now, let's turn to Darwinizing natural selection. Previously, I've mentioned that the idea of ecological scaffolds comes in the form of externalist explanation, forming a break from the usual internalist explanations of Darwinian properties and rising at a higher level in virtue of the interactions of parts, cells. As I've emphasized in section 2, despite the general reluctance among contemporary neo-Darwinian evolutionary biologists to consider internal causes as a source of evolutionary change, many have nonetheless drawn internalist patterns of explanation to avoid the circularity problem of explaining the emergence of Darwinian properties. One popular example for an internalist explanation of a transition in Darwinian individuality is the idea of co-option. In co-option, pre-existing traits of particles become relevant for the collective entity, turning it into a Darwinian individual in its own right. Black et al. Give the example of the colonial volvocene green algae, where groups can form by co-opting the retinoblastoma cell cycle regulatory pathway in the unicellular Calidomonas C. Hanschen et al. 2016. Similarly, they point to exciting work of the Radcliffe lab on snowflake yeast. Saccharomyces cerevisiae, butchering it here, able to generate collective level reproduction through co-option of the programmed cell death, apoptosis, to create propagules, thus showing that Darwinian properties can emerge from the interaction of parts. Nothing about this needs to be vitalist or mysterious. The point of Rainey's idea of ecological scaffolds is decidedly not to say that as a competitor to co-option explanations. It would be exciting to see where these experimental research programs on possible co-option processes will lead us in the next decades. We may start to think the actual evolution of multicellularity could have been a result of both co-option and ecological scaffolding, resisting a need fitting into either category. Here it would be a mistake to restrict ourselves to only one model. As I've previously argued, quote, for almost any aspect X of phenomena Y, scientists require multiple models to achieve scientific goal Z, quote end. But to restrict ourselves to co-option may force us to miss the important role of ecology. As Rainey would put it, we attempt to strip the problem to its bare bones, taking nothing for granted, and see how far we can get. Unfortunately, the debate often reflects a higher order commitment to kin selection and the co-option of almost clonal cells and to multi-level selection in the scaffolding approach. But these shouldn't be seen as mutually exclusive, such that co-option necessarily beats ecological scaffolding. This is easily recognized when we look at the very emergence of life, when there are no Darwinian units whose properties could become co-opted. Rainey's idea is even more useful outside of his work on multicellularity. It is at the origin of life as a process that ecological scaffolding provides us with a Darwinian view of how Darwinian properties emerge. It Darwinizes natural selection. Ecological scaffolding may provide us with a highly fruitful framework to develop plausible explanatory sketches of how natural selection could itself gradually emerge in an eco-divu feedback loop. While Black et al. Only talk in a few places about the transition to life from non-living matter, the idea of scaffolds I find a natural place within recent work on the origins of life. I will not go into any empirical details on this issue here, since I'm treating ecological scaffolding at a fairly general level as something that can explain the emergence of Darwinian properties in a non-circular yet Darwinian way. In order to Darwinize natural selection in this way, I will introduce a concept that I shall dub evolutionary scaffold, as a broader conceptual framework in which to understand Rainey's notion of ecological scaffolding. Drawing on our general definition of scaffolding, we can define evolutionary scaffold straightforwardly as follows. X is an evolutionary scaffold if and only if 1, X exogenously induces or supports the evolution of property Y and process system Z. 2, X vanishes from or becomes part of the system, while property Y and process system Z becomes endogenized. Importantly, this is not how Rainey sees ecological scaffolding. For Rainey, it is a scaffolding regardless of whether the properties induced by the environment become endogenized or not. He effectively gives up the second part of by definition, since endogenization need not be realized. But this is then returning to the overly expansive usage of the term scaffold as a mere environmental supporting relationship, a criterion that is notably weak due to its broadness, where simple models of the origin of life and multicellularity may idealize all environmental factors away, merely consisting of the replication of units. Almost all actual biological processes involve feedback between organism and environment. The real power of Rainey's idea of ecological scaffolding, I suggest, is thus not this environmental role per se, but rather the process of endogenization. To make this clear, it will be helpful to elaborate further in evolutionary context what is meant by endogenization. Borrowing the term from the economic modeling literature, where exogenous and endogenous variables are typically distinguished, the former are treated as given inputs, whereas the latter as outputs determined by the model. Okasha 2018 suggests that a similar distinction can be drawn in evolutionary models. Applied mathematicians switching from economics to biology may be surprised to learn that the role of endogenization has been given little attention in evolutionary models. Yet, this omission can simply be seen as an artifact of the modeling choices inherent to any evolutionary model. Some factors are taken as mere inputs, whereas others can be influenced by the model itself. In section 2, we saw that natural selection is often treated as a law-like ordering principle of biology. This largely owes itself to the procedural subsumption of more and more phenomena under the general explanatory scheme of evolutionary theory. Okasha 2018 describes this historical trend as a strategy of endogenization. Quote, it involves devising evolutionary explanations of four biological features that were originally part of the background conditions or scaffolding against which such explanations took place. Quote end. In the language of modeling, we can see this trend as the subsumption of phenomena under a more general model of evolutionary change. There are thus two senses of the term endogenization. One about a phenomena being subsumed, integrated into an explanatory program, the other about a phenomenon being endogenized over evolutionary time into the development of the organism. Both Wimsatt and Griesemer and Caporal et al. Briefly talk about the internalization of scaffolds as an important factor involved in the evolution of complex forms of organization, both in biology and culture, but unfortunately treat this as optional. Whereas these authors see such a process as an important role for scaffolds, I see endogenization of Darwinian properties at the very heart of what makes something an evolutionary scaffold. This distinction provides us with an interesting further question of how well the endogenization of variables into an explanatory model can serve as an adequate representation for endogenization of scaffolds in nature, such as Rainey's reeds, that I will further address in this section. Despite this ambiguity in the usage of the term endogenization, I expect that it will nevertheless be useful to think about our attempts at modeling the origins of Darwinian units and exogenous variables becoming endogenized. The explanatory reach of evolutionary theory has constantly pushed its limits, though this has often been criticized as too adaptationist. Yet, Okasha 2018 elegantly demonstrates that evolutionary theory has been extended to explain many phenomena, formerly considered to be outside the purview of, or at least obstacles to, Darwinian explanations, such as the origin of variation, biological sex, or anisogamy, altruism and population structure, niche construction, the genotype-phenotype map, and the origins of hierarchical organization. This is not to say that these phenomena have been fully explained in Darwinian terms, but rather that we are developing evolutionary models where these phenomena are treated as outputs rather than just take them for granted. These points help to illustrate what Rainey has in mind when he speaks of an endogenization of Darwinian properties. Internal factors, development, become more important with life becoming more autonomous from external forces in the process. In 2006, Okasha distinguished between the old approaches to the levels of selection question as synchronic approaches that treated hierarchical levels as given in the explanation of adaptations on that level, whereas he considered the growing evolutionary transitions literature to constitute a diachronic approach to the levels of selection question. Here, the levels of selection itself is explained through recourse to Darwinian explanation, which, as Okasha 2018 points out in his recent paper, can be understood as the endogenization of the, quote, hierarchical organization itself, quote end. Hierarchy is no longer seen as a necessary exogenous factor in evolutionary models. This binary way of describing the growing major transitions literature can be misleading, however, since it may be interpreted as suggested that we have already subsumed these phenomena under the scope of the principle of natural selection. Partially, this is due to the origin of the endogenous-exogenous distinction that Akasha draws on from the modeling literature in economics. In economics, either variable is an input of the model or it is an output. But in nature, we find gradations in endogenization as a natural phenomenon that should not merely be understood as a feature of our models and explanations. It is a real phenomenon in nature, and it is the very origins of this endogenization process that is the origin of development that we should pay attention to, since it is here the natural selection emerges in an eco-devo feedback loop. It's because of this that ecological scaffolding ought to include endogenization in the sense of ecological properties becoming organismal ones. That is the second component of my scaffolding definition, in order to turn it into from a mere metaphorical notion into an explanatory concept. Much in the above discussion of Rainey's work is focused on reproduction, but other Darwinian properties face the same problems. For discussion of heredity, see Dolcier et al. Nevertheless, as Okasha 2018 describes it, Darwin's worries about the origins of variability in populations have been solved, and to some extent this is certainly true. We no longer treat variation as a black box. We have come up with both short-term and long-term macroevolutionary explanations for why variability itself can be explained as an adaptation. Indeed, this work elegantly develops with work on evolvability, see Pigliucci 2008. But debates about the role of evolution or rather natural selection and emergence of a phenomenon rarely focus on the question whether or not natural selection is part of the story. Let me just drink some water. The question is how important natural selection is in the explanation of a particular phenomenon, as opposed to other factors such as physical or chemical constraints and the possibility of perfect replication. This is why the abbreviated Van Balen quote in Black et al. 2020 about development being controlled by ecology must not be misunderstood as suggesting that we need either developmental or an evolutionary explanation. Rather, Rainey defends the idea that development and evolution are two sides of the same coin. They are both in control of each other. There's no interesting thesis here about one beating the other, and we need to pay more attention to the evolution of development. As Griesemer 2000 noted, development has long been a weak link in evolutionary theorizing and ought to be integrated in our thinking rather than idealized away. Similarly, Van Balen maintained that despite the possibility of thinking about evolution in terms of ecology controlling development, quote, neither area has figured importantly in evolutionary theory since Darwin, who contributed much to each. Quote end. And it is this more important point by Van Balen that makes Rainey's ecological scaffolding framework such an interesting proposal. Through integrating development, evolution, and ecology in an eco-evolutive feedback loop. Even at the longest possible timescales, it suggests that development is a factor that cannot be idealized away. Rather, it is a process that underlies the very endogenization of Darwinian properties in their own right. Ecological scaffolding explains the very origins of internalist developmental explanations without taking anything for granted. I'm thus not here interested in claiming that development controls evolution through the endogenization of Darwinian properties. There is as much truth to the statement as there is to Van Balen's. Indeed, Okasha warns us not to confuse the widespread prevalence of endogenization in evolutionary biology with a reductionist view of natural selection as a universal asset, see Dennett. Even if we want to treat natural selection as something like a first principle of science, we can defend a more nuanced view that likewise makes us use the idea of endogenization. Block quote by Samuel Okasha here. The alternative is this. It is not the core Darwinian principles themselves that bear the explanatory burden in evolutionary biology, but rather those principles as they operate in specific biological settings, in the presence of additional contingent biological features. Quote end. Naturally, there's a myriad of ways background features can play a role in specific explanations that make use of natural selection. If these can in turn be explained through natural selection, it is tempting to buy into Van Walen's dictum. But Okasha 2018 rightfully maintains that even if we can provide an evolutionary explanation for the background features of a particular phenomenon, we want to explain this does not entail that a phenomenon can be explained without an explicit appeal to the background features. What is important for the purposes of this paper is simply to recognize that background assumptions often serve as a scaffold in biological theorizing, only to later become part of a Darwinian explanation. Let me read out this important footnote here. Consider an egalitarian marriage of the 21st century. Historically, marriages were sometimes so jokingly described as a man being chained by a woman, suggesting that women are in control of their husbands. But this would be a gross misrepresentation of the patriarchal and sexist power structures we found in marriages in the past, and unfortunately, many in the present. Such statements have largely been abandoned, or at least come to be scrutinized due to the sexist nature. In an egalitarian marriage, however, it need not be the case that either partner has power over the other. Unless we want to define the term control as whoever has more causal power, we can legitimately say that each partner has control over another. And so it is with development and natural selection. We do not have to defend a view in which one side must win. But in an ecological scaffolding, as I aim to more narrowly define it here, it is not these background conditions of an explanation that become endogenized, but the phenomenon itself. By understanding ecological scaffolding as an externalist attempt to endogenize Darwinian properties, we can tease apart this duality in Rainey's discussion of ecological scaffolding between what can be understood as an explanatory scaffold and what I've called an evolutionary scaffold. This distinction is subtle but important. On the one hand, ecological scaffolding can be understood as an externalist as opposed to internalist mode of explanation, in which Darwinian properties are induced by Darwinian means in a non-circular fashion. Here we rely on natural selection, environmental filtration, as an explanatory scaffold for explaining the origin of Darwinian properties. We subsume these phenomena in a Darwinian model of life. On the other hand, the environment itself is an evolutionary scaffold, a genuine scaffold in nature that induces Darwinian properties that are then able to become endogenized across evolutionary time. One is a tool for coming up with new explanations for Darwinian individuals. The other is a natural phenomenon that we are trying to make sense of here. In trying to understand the role of scaffolds in nature, we can also recognize something of a reverse process of evolutionary scaffolding, in which Darwinian properties become exogenized rather than endogenized, with organisms losing autonomy and becoming more dependent on special ecological conditions. Such an idea has been hinted at in Godfrey Smith's discussion of viruses that, while brief, can be elegantly co-opted here. A block quote: simple reproducers need not be the lowest level reproducing entities in our hierarchy, however. A third category I will call scaffolded reproducers. They might even be called reproducees, or at least many of them could. These are entities which get reproduced as part of the reproduction of some larger unit, a simple reproducer, or that are reproduced by some other entity. Their reproduction is dependent on an elaborate scaffolding of some kind that is external to them. However, these entities do have parent-offspring relationships. Hence, they form lineages or family trees. Quote end, Godfrey Smith, 2009. Viruses straightforwardly use other organisms for the continuation of their own life cycle. They depend on the reproduction of other living entities in order to facilitate their own reproduction. Without the scaffold, the life cycle is stuck, an evolutionary dead end. It is not, however, evolutionary scaffolding, as I constrain the term. While viruses did evolve via other reproducers, they did not discard or endogenize them. Only if a virus were to evolve the ability to reproduce without the need to hijack the reproductive capacities of other organisms would there be a case of evolutionary scaffolding. It may thus be worthwhile to draw a distinction to developmental scaffolding, which better describes Godfrey Smith's notion of a scaffolded reproducer. Nevertheless, in thinking about evolution, something very much like a reverse ecological scaffolding process must have been at the origins of virus lifestyle. and perhaps more generally at the evolution of parasitism, when autonomy was lost. Hence, we should recognize that the role of scaffolds in evolution can go in two ways: endogenization and exogenization. In the former, scaffolds become endogenized or discarded, that is, or e.g., the origin of life and plausibly the origin of multicellularity. And in the latter, organisms introduce scaffolds into the life cycles that make it evolutionarily viable to exogenize Darwinian properties such as reproduction, e.g., a virus. These two should not be confused, and it is for this reason why the scaffolding metaphor in the latter is bound to be misleading, since there are two quite distinctive processes going on. Perhaps a better way of distinguishing the two processes would be between endogenized scaffolding as a process of autonomization from external forces and the exogenization of development as a loss of autonomy, reliance on external factors. Nevertheless, in both it is the role of the ecology that is the crucial key to understand evolutionary dynamics these systems take, and we can see my narrow concept of evolutionary scaffolding as enabling us to distinguish these two directions evolution can take organisms. I hope that my refinement of Rainey's idea of ecological scaffolding as a process of endogenization will help to highlight the importance of this simple fact. Natural selection is an ecological process and should therefore be studied in terms of the external ecological scaffolds that give rise to the eco-devo feedback loops of life. Before we move to the conclusion, however, I hope that this section has provided something of a proof that Caporal et al. Were right in their prediction for the usefulness of a developed concept of scaffolding. Quote, a more highly analyzed, developed concept of scaffolding will highlight the role of temporal and temporary resources to development broadly conceived across concepts of culture, cognition, and evolution. Quote end, Caporal et al., 2014. Section 5, Conclusion and Further Avenues for Research. The theory of natural selection is the most impressive theoretical development in the natural sciences. No other theoretical framework has achieved the striking explanatory breadth and growing list of successes of this basic Darwinian idea. Yet the major transitions have continued to resist this Darwinian imperialism. The aim of this article has been to offer the first philosophical analysis of Rainey's idea of ecological scaffolding as an ambitious attempt to once more push the boundaries of externalist Darwinian theorizing to encompass the major transitions, including the very origins of life. Firstly, I have argued that Rainey's work constitutes a beautiful example for the possible fruitfulness of collaboration between philosophers of biology, theoreticians, and experimental biologists, and it provides both a general theoretical framework and an empirically testable hypotheses that can be implemented in the lab with plenty of possibilities for feedback between both. Collaboration between philosophers and evolutionary biologists is, of course, not a new phenomenon. See Lloyd et al. For a particular elegant case, they have worked together on the nature of evo-devo and on how theoretical field and lab work interacts in practice. Naturally, this is far from an exhaustive list, and many more examples could be given once we extend our view to experimental evolutionary ecologists such as Robert Brandom and Yanis Antonovics. But what I hope this paper has shown is that philosophers of biology would do well to keep up to date with this exciting new work in experimental biology, addressing old philosophical problems with exciting new experimental tools. In this, I can only echo a recent critique by Prideaux, 2017, block quote, "Clearly, evolution offers one unifying framework for all biology, and some aspects of evolution are highly theoretical. But this should not hide the fact that evolution is also a key experimental component. Recent major advances in evolution have come from studies in experimental evolution, such as those of Richard Lenski, Michael Travisiano, and several others. An exclusive focus on the theoretical dimension of evolution might lead philosophers of biology to miss the importance of these experimental approaches to evolution." Quote end. It was Prideaux, 2017. Secondly, I've tried to offer an analysis and explication of ecological scaffolding, an idea that, while highly useful and fruitful for the illumination of old biological problems in the major transitions literature, could benefit from further refinements by philosophers of biology. While usage of the term scaffolding has exploded in recent years, it has often been used in rather imprecise, vague, and merely metaphorical ways. Here I've attempted to give both a precise yet general definition of scaffolding and evolutionary scaffolding that restricts Rainey's idea of ecological scaffolding to only those circumstances in which exogenously imposed properties come to be endogenized, since it is here that we come to observe the dawn of development. In addition, I've argued that this emphasis on endogenization allows us to distinguish a reverse scaffolding process, with organisms becoming less autonomous, that must have occurred during the evolution of viruses and other parasitic life cycles. Future work may attempt to model the adaptive dynamics leading to exogenization and endogenization. The greatest strength of the inherently Darwinian explanatory strategy of scaffolding may indeed lie outside of its original application, that is, at the origin of life, when there are no lower-level units with Darwinian properties that could be co-opted. It is in this sense that the very origins of natural selection may very well have required something like a complex ecological scaffold. Indeed, this recognition may help us to explain why the origins of life research has failed to make much progress, a partial neglect of ecology. While one may legitimately reply that the study of the chemical conditions for life has been about the ecological conditions of proto-life forms, this work does seem to have been dominated by an internalist mode of explanation, which focused on the minimal conditions of living systems without a role for ecological scaffolding. This is why I've titled this paper Scaffolding Natural Selection. The origins of life may require appropriate ecological conditions in order to scaffold Darwinian properties onto non-living systems in something like an original eco-devo feedback loop, in which natural selection itself gradually emerged alongside the process of life itself. However, I have also argued that we should resist the temptation to label any environmental support found in nature as environmental scaffolds, as this would make the explanatory concept hollow. Scaffolding can indeed be further developed into a genuine scientific concept with explanatory power, but only if we restrict it, as I've argued, to processes that contain the very origins of the endogenization of Darwinian properties. Further, directions that deserve exploration are the connections between development, ecological scaffolding, and natural selection. While this paper is aimed to address these issues, I've only taken some of the first steps, and there's much further work that needs to be done. Using the framework suggested here, many puzzling problems about evolution of multicellular organisms and even the origin of life may come to be illuminated. The scaffolding of natural selection offers an exciting new research area within experimental evolution, and it is likely to play an important role in future research on the major transitions. Unfortunately, it has been common among evolutionary biologists to ignore the role of development at the timescale of the major transitions. But this is a mistake. In evolutionary scaffolding processes, whether endogenizing or exogenizing, much hinges on the development of complex life cycles. I expect that future work inspired by Rainey's experiments will show the need to distinguish between what we may call developmental scaffolding as a process within a life cycle and evolutionary scaffolding as a process across life cycles, and how these forces can pull in opposite directions in the cases of multi-level selection. Unfortunately, a proper treatment of these problems will be a task for another day, though see Griesemer for a set of interesting ideas. Nevertheless, this need to distinguish different scaffolding processes only further emphasizes the need for greater conceptual clarity. Finally, I hope that there's at least some kernel of truth to be found in the analysis I've offered, and that it will be useful to philosophers, experimentalists, and theoreticians alike in our joint goal to further progress our understanding of evolution. To conclude, scaffolding is a natural phenomenon, one that plays a myriad of underexplored yet important roles in evolution. We can be Darwinians about the Darwinian process of natural selection itself, thus once again broadening the scope of natural selection as a first principle of biology, as expressed in Theodosius Dobzhansky's famous dictum that, quote, nothing in biology makes sense except in light of evolution, quote, end. All right, I hope you enjoyed this paper. And what can I say? Yeah, one of my favorite papers. But it's very long. 15,000 words. I was lucky that journal editors didn't just say, well, that's just far too long. Part of the problem was the review comments, which required for the paper to get even longer, and as that sometimes happened. But I think The paper is very strong. Make a good argument here for how we might reconceptualize the very origins of how natural selection may get started. And this way may well become the basis of a longer book treatment on these issues. So I hope you enjoyed this episode, and um, maybe you'll see me again in another episode. So enjoy your week.